السلام عليكم ورحمة الله وبركاته، ازي حضراتكم جميعا. أحب أشكر الدكتور عصام وأستاذنا الدكتور خالد الباسل والدكتور راشد حسن على الدعوة الكريمة وأساتذتي في المنصة. I'm Zakrya Ibrahem, lecturer of tropical medicine in Shams University, and this is a talk about circadian pancreatic clock dysfunction. عنوان غريب وكثير يعني مش كتير قابلنا العنوان ده، وانا قعدت بصراحة من ساعة ما قال لي الدكتور راشد ادور فيه بس عشان نحاول نفك طلاسمه مع بعض ان شاء الله. Starting from the basic definitions, what do we mean by circadian? Circadian comes from the Latin word circa, dies or dime, which means around the day. And circadian rhythm refers to the cycle, 24-hour cycle, which involves the body's biochemical and physiological process. And this represents the internal biological clock. The importance of the circadian rhythm is that it governs many of the biological functions, including sleep and the government of the hormone secretions like the melatonin and cortisol, the metabolism and the mental health, and even the immune response can be governed by the circadian rhythms, as we shall see. Here's a 24-hour cycle which denotes the different physiological process occurring in a timely manner, like the best coordination at 2:30, like the peak of melatonin secretion starts at 9 p.m., like the deepest sleep at 2 a.m. So these different physiological processes occurs in different time intervals and reach their peaks at circadian maneuver. The study of the biology related to time, which is chronobiology, started since 1970s and deepened our understanding of the circadian systems. In the center of the what we call peripheral oscillators or peripheral clocks is the pancreas, because it is both endocrine and exocrine organ, as we shall see, and the circadian rhythms depends centrally on the suprachiasmatic nucleus, and peripherally, essentially every cell may act as a peripheral oscillator of circadian rhythm, but the main peripheral clocks include the heart, immune system, the pancreas, and the liver. The interplay and the crosstalks between the suprachiasmatic nucleus and these peripheral oscillators give the circadian rhythm characteristics, and this paves the way for interventions because disruption of the circadian rhythm has been associated with different metabolic consequences and even cancers. And subsequently, if we are targeting time or circadian molecules, stabilizers, or analogs, this shall help with the treatment or even prevention of these different metabolic derangements. And starting from the pathophysiological aspects, the central master clock is the suprachiasmatic nucleus in the hypothalamus. The suprachiasmatic nucleus in the hypothalamus represents the master clock of the circadian rhythm, and this is influenced by the light-dark circle, and together with the food intake, and by itself through neuroendocrinal mechanisms affect the peripheral oscillators like the pancreas. The circadian rhythms depends not only upon the external stimuli, but even endogenous characteristics like gene polymorphism, age, and sex plays a role in the circadian rhythm. What if this circadian rhythm had misalignment or disruption? It will lead to metabolic syndrome involving obesity, elevated LDL, low HDL, and the development of type 2 diabetes thanks to insulin resistance and beta cell dysfunction. Here is the algorithm. This demonstrates the center clock, suprachiasmatic nucleus, which interacts with the peripheral clocks, leading ultimately to mediating the different metabolic functions. And the external factors, namely the change in the light cycle and the food availability, represent the extrinsic factors that may affect this circadian rhythm. The pancreas, because we are talking about the circadian rhythm disruption in relation to the pancreas, Professor Dr. Khalid Al-Basil demonstrated the basic structure of the pancreas acting as exocrine organ and endocrine organ through the islets of Langerhans. And this represents the different pancreatic contributions to the circadian rhythm mediated via the different hormones, insulin, glucagon, somatostatin, and amylin, which by themselves affect the different metabolic processes and even the brain, like we shall mention. This interaction plays a paramount role in the blood glucose hemostasis, which depends upon the crosstalks of the different cells of the pancreas. Alpha cells secrete glucagon, beta cells secretes both insulin and amylin, gamma cells produce pancreatic polypeptide, and delta cells secreting somatostatins. Insulin itself, its secretion is controlled by local mechanisms and distal signals coming from the intestine, stomach, and other hormones. In the arcuate nucleus of the hypothalamus, insulin influences both anorexigenic and orexigenic neurons. These neurons are the neurons associated with the sense of hunger and the sense of satiety. And insulin, through the affection of these neurons, can control the response, and this response is mediated via what we call the pro-opiomelanocortin, pomic and cart cocaine amphetamine regulated transcript. These are activated by insulin, and by their activation there is reduced food intake and increase in the energy expenditure. On the other hand, neuropeptide gamma and A gout related peptide are inhibited by insulin, further decreasing food intake. Amylin reduces food intake by inhibiting the neurons responsible for hunger and mediates through slowing gastric emitting and inhibition of the gastrointestinal secretions and inhibition of the postprandial glucagon secretion, and somatostatin acts by inhibition of the insulin, glucagon, and the different pancreatic and digestive enzymes. Also, circadian rhythm plays a role in insulin resistance. Insulin resistance has endogenous factors and exogenous factors. Endogenous factors is demonstrated in the genetic makeup in persons with insulin resistance, and also immune response may play a role in the insulin resistance via the cytokines released by macrophages that promote adiposity and insulin resistance. These responses, this food intake and the immune reaction are all influenced by the circadian rhythm, altering this process based on the time of the day. And that's how circadian rhythm shall influence insulin resistance. So in a conclusion, the blood glucose and the insulin resistance are controlled both centrally and peripherally, and the affection is associated with the development of impaired metabolism, type 2 diabetes, and experimentally, the suprachiasmatic nucleus upon ablation caused disruption of the glucose metabolism and led to irregular hormone levels. Other hormones that are acting in a rhythmic fashion as cortisol, which follows a daily rhythm, and the peak occurs after waking. The gastroinhibitory peptide and the GLP-1 also show circadian rhythms, and disturbance of which is tied to obesity and type 2 diabetes. Ghrelin, named as hunger hormone, also its secretion follows circadian rhythm and inversely related to insulin. What about the molecular basis of this circadian rhythms? At the molecular level, there are genes that control this circadian rhythms, namely BMAL1 and BMAL2, referring to brain and muscle aryl hydrocarbon receptor nuclear translator, and circadian locomotor clock, and cry, cryptochrome, and period. All the names are quite difficult, but these are genes controlling the circadian rhythms. And as we shall see that these molecules has been seen in the development of the pancreatic ductal adenocarcinoma, as Stefano and Ital found a significant downregulation of these key circadian clock genes, namely BMAL1, CRY1, 2, BER1, 2, 3, in this type of malignancies. Also, in cases of chronic pancreatitis, they found that 1/3 of patients with the signs of what we call desynchronosis or the altered circadian rhythm, those patients with chronic pancreatitis had altered cytokine secretion profiles, and this increases the likelihood of developing pancreatitis. Also, they have been associated with disruption of the rhythmic secretion of melatonin, which both mediates the circadian rhythm and also inflammation. The single nucleotide mutations in these key clock genes has been associated with the increase of likelihood development of obesity, metabolic syndrome, and type 2 mutations. And the polymorphism, the nucleotide polymorphism in BMAL1 has been linked to hypertension, type 2 diabetes, and gestational diabetes. The polymorphism in the BER2 gene was associated with binge eating and the weight gain. Also, the polymorphism in the melatonin receptor 1B gene has been linked to higher fasting glucose level and the destruction of the beta cells and increasing the risk of developing type 2 diabetes. These are the studies that studied specifically the nucleotide polymorphism among the different genes involved in the circadian rhythms and the association with the different clinical conditions. We can induce this modulation of the circadian rhythms via different mechanisms, namely forced desynchronization. We force this misalignment in the circadian rhythms via the changing or altering the feeding pattern and the sleep-wake cycles, making them longer or shorter than 24 hours, aiming at disruption the center from the peripheral oscillators. And these are the factors that may affect the circadian rhythms. We can change the light cycles, we can change the sleep pattern, and we can cause sleep deprivation, or change the food, whether liberal food intake or food restriction, just to make alteration or misalignment of the circadian rhythms and study the consequences. There has been some studies, whether on mice or on human being, to demonstrate these effects. In healthy lean men, the bright light effect on triglycerides, whether fasting or postprandial, was high. The exposure to bright light caused increase in triglycerides both fasting and postprandial. And likewise, the condition in obese men with type 2 diabetes. The bright light effect on glucose on this cluster of patients, obese men with type 2 diabetes, has been associated with improved fasting and postprandial glucose. Also, there have been studies on the food timing affecting the circadian rhythm among mice. All these are studies to study or to highlight the effect. One factor that may affect the circadian rhythm is the shift work. Shift work, the night shift workers versus the day shift workers have higher postprandial glucose, insulin, triglyceride levels than the shift day workers. And the issue of shift work by itself increases the risk of developing of type 2 diabetes by 9%. And the rotating shifts have higher risk than constant shifts. Jet lag represents the feeling of tiredness that the person gets while traveling a long distance because of time differences. And social jet lag represents the temporal discrepancy between person's sleep pattern on working days and on non-working days. Unfortunately, most people today experience social jet lag due to the mismatch between the biological and social schedules. Persons with jet lag may develop mood changes, cognitive impairment, appetite loss, GI disturbances, and even with chronic jet lag, they may develop cancer and cardiomyopathy. Social jet lag with one more than hour of social lag raises the possibility of prediabetes and diabetes by 75%. This paves the way for what we call chronotherapy. Chronotherapy means the administration of drugs at optimal times of the day to maximize their benefit. This has been seen in statins. Statins are typically given in the evening. They inhibit MGK-OA reductase, which peaks at night. Bromocriptine, acting within 2 hours of waking, reducing the hypothalamic dopaminergic drive. Metformin, there have been no clinical trials on the optimal timing, but many studies should follow to demonstrate the role of chronotherapy. As a matter of summary, the circadian rhythms are shaped by external factors: sleep, wake cycles, light exposure, eating times, and activity, together with internal factors: age, genetic chronotype, and the time of the day the person is naturally alert. And there is no standardized scoring system to assess exogenous circadian influences, and yet no genetic screening to identify the genetic susceptibility to circadian disruption. As a matter of potential future tools, there could be personalized disease management based on individual circadian profiles with optimization of drug timing to maximize its efficacy and side effects. And insight from these studies could inform simple lifestyle intervention together with the molecules that may affect the circadian rhythm. And thank you.
شكرا جزيلا بروف زكريا ابراهيم، شكرا جزيلا على المحاضره القيمه. Any questions يا فندم؟ طيب تمام.
شكرا يا دكتور، شكرا. مسك الختام بنكرياتيك جروب طبعا الاستاذ الدكتور وليد احمدي، بروفيسور زاكز يونيفرسيتي. طبعا معظم الحضور هيسال على الموضوع الشاق اللي الاستاذ هيقوله لنا